So this is Unit 4, Part 2 of the immune system. Today we're going to be looking at the specific immune system. Before we do that, we need to define what an antigen is. An antigen is a surface protein on a pathogen, and this causes the specific immune response. And so this surface protein is something that the body recognizes either to be a new invading pathogen or one that your body has dealt with before. Either way, there is then a specific response that is going to be initiated against that pathogen. There are uh, proteins on macrophages and other cells that can read those antigens and then kind of figure out how they're going to initiate the response. If it's something that you've never come in contact with, then that response is called a primary immune response. This is, again, your first encounter with the pathogen. It generally takes a couple of weeks, 10 to 17 days, for the right antibody to be made in order to deal with that particular pathogen. Then if you come in contact with it again, because you have cells called memory cells, and they remember things, makes sense, that secondary immune response is a lot shorter because your body knows how to deal with that. Those immune cells proliferate into the body and into the blood, and they deal with that infection very quickly. Now, as we are looking at the specific immune response, here it is, and you can tell that it could be complicated. There are two sides to this. Now, one thing that's important is that these are both happening, and you can see this. The antigen is, you're exposed to the antigen. The macrophage eats it. It says engulfed by the macrophage, and then it's going to present that antigen. Remember the MHC class 2s that we talked about in the first video. It's going to present that antigen, and then that's going to cause both sides of this specific immune response to be initiated. On the left, you have the humoral immune response, or antibody-mediated response. This is happening in the fluids. Humoral means fluid, and so this is happening in the blood. And then you're going to have the cell-mediated immune response. This is mediated by actual cells. And so whereas on the left, on the humoral response, this is mediated by proteins called antibodies, which we'll talk about shortly. The right side, the cell-mediated response, is mediated by actual cells. And so let's look then first at the humoral response. So the macrophage is going to eat an antigen, and then it is going to present that antigen using the MHC class 2. Now, how does it get the T cell to come up to it? Well, it releases a chemical called interleukin 1. And so interleukin 1 is released, and that interleukin 1 causes this other cell to come up to it called a helper T cell. And the helper T cell is a very important cell because it is going to analyze that antigen that has been captured by the macrophage. And that is going to cause the helper T cell then to call to action both the humoral response and the cell-mediated response. So again, macrophage eats a bacteria or virus, breaks it down, has the antigen, says this is the thing that I just ate. The helper T cell is called in by interleukin 1 and analyzes that antigen, and now the helper T cell is going to initiate both sides of the immune response. On the left, it initiates the humoral immunity by calling to action the B cells. Now, the way that he stimulates the B cells is by releasing a chemical of his own called interleukin 2, just IL-2. That's going to stimulate the B cells, and the B cells are going to do two things. The B cells, some of the B cells are going to be converted into a cell called plasma cells, and those plasma cells are going to begin producing antibodies. Here's an antibody on the left here. This is a quaternary protein, and you can see it's made up of several structures here. You see the disulfide bonds, and there's heavy chains, light chains. More importantly, I want to focus here. There's the C region and the V region. C and V stand for very easy things. There's the constant region. This is going to be the same on any antibody. This is kind of like when you go to Lowe's and you're looking for a garden tool. All the garden tools have something in common. They all have a long wooden handle. And so this is the long wooden handle of the antibody. Now, the business end of the garden tool has a different thing on it depending on what you need. It could be a shovel, it could be a rake, it could be a hoe, it could be lots of different things. And so that is the V region, the variable region of the antibody. The variable region of the antibody is going to be different depending upon the type of antigen that was received. And so this is why it takes the immune system a while to figure out pathogen during the primary immune response. What type of antigen needs to be looked for? I need to make a tool that fits this specific antigen. And so when that is found, the antibodies then proliferate into the blood and they find the antigens. They find these bacteria or the viruses or whatever, and they bind to them. And what that does, as you can see, is this pathogen, when it has these antibodies all over it, it's going to be unable to connect to your own cells, and it's like little flags, and those little flags call in the macrophages. The macrophages come in and gobble them up. And so again, this isn't initiated by, well, the B cells produce these, but the antibodies are what are actually going out and doing the job. And these antibodies, again, are little proteins that attach themselves to a specific kind of pathogen. And that pathogen, again, that antibody has a variable portion that binds to only one type of thing, and that is that specific pathogen that has been uh, eaten by the macrophage. Now, plasma cells make those antibodies, but also there's going to be some B cells created that are called memory B cells. And the memory B cells are cells that are stored away for future reference. Basically, the next time that you come in contact with this pathogen, then the B cells are going to know exactly what to do. They're going to be producing the antibody that they know exactly what to do with, and that immune response is going to be much quicker. It's kind of like playing a team for the second time. If you are playing a team for the second time, you've already watched film on them, you already know what to expect, and so you can formulate your defense easier than you could have the first time you played them. So that's the humoral side. Let's look at the cell-mediated side. Same thing. You have interleukin 2 that is released by the macrophage. Helper T cell is going to be stimulated by that. It's going to analyze that antigen, and it is going to release interleukin 1. And interleukin 1 is going to call in a group of cells called cytotoxic T cells. Sometimes you'll see them called killer T cells, which sounds more fun. Either one is acceptable. And what these cytotoxic T cells do, very similar. Notice there's going to be two types. There's going to be ones that are actively doing their job, and then there's going to be ones that are memory, that are going to remember the thing that you're supposed to go fight against. And so again, the helper T cell is going to, this is what you're going to go fight against. The cytotoxic T cells are now going out to look for infected cells. How will they know which cells are the infected ones? Well, those infected cells are going to be holding out MHC class 1s. This is the thing that's killing me. And then the killer T cell goes in and releases a protein called perforin. Perforin tells us what it does. It perforates those infected cells, causing them to lyse or to explode, and then preventing further infection from being spread. And this is very similar to, if you're firefighting in the forest, you're going to start fires in some places to burn off sections of the forest in order to keep the fire from spreading into those sections and hopefully stave off the fire. And so this is another way of doing that. It keeps the infected cells from producing more of the pathogen, particularly in a viral infection, and this leads to your immune system being able to deal with those. And so back to this big picture, you can see helper T cell is a very essential portion of the immune response because the macrophage eats that antigen, shows it to the helper T cell. Helper T cell stimulates both B cells and killer T cells to release antibodies and create these killer T cells. to go out and to do the work of the specific immune response.